So, hello everyone, I'm Maxime. It's a pleasure to be here. And as Jean-François said, I'm coming from France, from the Ecole Polytechnique, to talk to you about how we can use deep generative models to do semi-supervised learning while being robust to label errors. So, yeah, a quick word about the team that worked on the project. So there was Jeffrey, who's a postdoc at Berkeley, Romain, a PhD student there, Mike and Nir, two professors, and finally, last but not least, Edward was a fantastic co-worker of mine at Berkeley. So the model we're going to see today is built up on the framework of variational autoencoders. So a quick review of this framework. Let's say we want to embed our data in a low-dimensional latent space, Z. And for computational convenience and tractability, we take a prior on the distribution in the latent space that we're supposed to be normal, N0I. And our goal is to find the mapping from the latent space to the observation space that maximizes the probability of our data given the model. The problem is that this probability is not tractable. So one way to bypass that is to use neural networks to approximate the posterior distribution on the latent space, P of Z given X. And that allows us to derive a lower bound that we maximize in order to optimize our model. And we will refer to the lower bound as the ELBO. All right, so the variational autoencoders works very well in an unsupervised setting, and it's natural to extend it to do semi-supervised learning. So the state-of-the-art variational autoencoder model for semi-supervised learning is called the M1 plus M2 model. So it works is that, so you get some unlabeled data that gives you one part of the ELBO. You've got labeled data for which you condition on the observed label. It gives you a second part of the ELBO. And you have to extend this ELBO to give it a supervised feedback that is a classification error that is not motivated by the Bayesian specification of the model. And you have to do it in order for it to work. So there are two problems, two issues with this model. The first one is we know that in real life, labels are most of the time noisy. And this model performs very well when the labels are correct, but is not necessarily robust to noisy labels. And also, we'd like to know if we can get a probabilistic interpretation of this extension of the ELBO. Because the strength of the variational autoencoder framework is precisely to have a probabilistic motivation for every term that you optimize. And we'll see that actually those two questions are linked. So what we wanted to do was to use the strength of Bayesian modeling to integrate our knowledge about the fact that there can be errors in the labeling process within our model. So what we did was we looked at the generative model of the M1 plus M2 model, and we said, okay, here the observed label and the label that generated the data is the same. So if you suppose that there is an error process between the true label and the observed label, this is not modeled here. So what we did is that we built a model, the MVIE, that stands for mislabeled variational autoencoder, where the generative label that generates the data and the observed label are different nodes. And the observed label is actually a random variable that's parameterized by the generative label. So when you derive the ELBO for the MVIE, the major difference with the standard N1 plus N2 model is that you get naturally a classification error. So there's an expectation with regard to the latent variables. Then the posterior distribution on the true label, that's a weight, so the log P of y prime knowing y, that is defined by a matrix that has a size number of classes times number of classes. And actually this matrix defines how noisy the observed labels are. So we can have different choices for this matrix. The first one, that's the most simple and the most obvious, is to say, okay, we're going to model the error process so as being dependent only on the fact that the true label and the observed label are the same. So this is what we call the global error case. Or you can do something that's a bit more broad and say that the error process when you label the observed label with the true label is a function of y and y prime. So one intuition between the classification error term that we saw is that, okay, let's say that you're training your model on n-written digits. And you've got one data point for which the true label is a 7, but there was a mistake in the labeling, and the observed label that you get is a 1. As you specified to your model that it's very likely that you can get errors between 7 and 1s, this is going to be there is going to be an incentive for the model to not take too much into account the observed label, and that will give it the flexibility to base itself more on the features of the data point to label it correctly. On the other hand, let's say that you've got one of your data point that's a 5, and the observed label is correct and is also a 5, but at the beginning of the training, your model classifies it as a 6. As you specified in the Bayesian modeling that it's very unlikely that you get errors on 5, there's going to be a huge incentive for the model to label it correctly. And so throughout the training, it's very likely that the prediction for this digit will switch from 6 to 5. So this model seems fine. We have reasons to think that it can be robust to error in the labeling process. But we still haven't answered the second question, which was, can we give a probabilistic interpretation of the extension of the ELBO in the M1 plus M2 model? And actually, when you look at the global error case, so you can parameterize it by a parameter epsilon that symbolizes your uncertainty on your labels. And when you compute the ELBO in this global error case, you realize that you get the same ELBO as you got in the M1 plus M2 model, and that you actually have a one-to-one mapping between this parameter epsilon and the hyperparameter alpha that you had in the M1 plus M2 model. So what that tells us is that we can actually reinterpret the M1 plus M2 model as modeling uncertainty on the labels, but as being uniform on all classes. And with a value of alpha, the hyperparameter, that translates the belief on the uncertainty in the labeling process. So now we're going to see an application of this model in single-cell genomics annotation, and more specifically in single-cell RNA sequencing annotation. So first, just a quick explanation of what is single-cell RNA sequencing and why we do machine learning with it. So it's a new technology, so it's a big breakthrough in biology that allows us to get noisy measurements of gene expression at the single-cell level. So what that means is that we've got n samples, where each sample is a cell, and for each sample we've got G features, and each feature is a gene. So that gives us data which is n times G matrix, where each entry of the matrix is for a certain cell and for a certain gene, the count of the number of times the gene was expressed within the cell. And this, the state-of-the-art for single-cell RNA sequencing modeling actually relies on deep generative models. So for instance, the single-cell variational inference framework that we'll use in the rest of the experiments. And what they do is that you get a latent embedding of the data. You use that latent embedding to perform clustering. So in our case, clustering, classification corresponds to finding out cell types. And then you also use it to do hypothesis testing, which is detecting characteristic genes. And so that works pretty well. But the next step, what we wanted to do, was to be able to use some labels on the data to annotate a whole data set. So to work in a semi-supervised setting. And the problem is that those data sets, they come with only partially manually annotated labels that are very prone to errors. So why is that? The first thing is that to get those labels, you do a heavy pre-processing. So feature selection, which is gene sorting, sample selection, which is cell selection. After this heavy preprocessing, you go through a phase of dimensionality reduction, for instance, through PCA, and then use various clustering algorithms to label the cell types. And this very complex process can lead to errors. So the experimental setting we chose was to work with a data set that comprehends 3,000 cells from a mouse brain that were manually annotated by biologists. So we've got 7 different cell types. And here you can see a latent embedding by the unsupervised method. And if you look closely at the clusters, there seems to be a lot of points that are misclassified. So that was what motivated the work in the first place. And what we do is that we focus on only 2 cell types for the sake of simplicity. And so we take those 2 cell types and we say, okay, we know there are some classification errors, but we want to use those labels as ground truth to try out our model. So what we do is that we artificially corrupt the labels with a magnitude that's far greater than the one that is already present in the labels. So for instance, for the blue dots, with 20% of chance, we turn them into orange dots. And for the orange dots, we choose various corruption probabilities. And then we train our model on this corrupted data set, and we look if it is able to find back the true labels. And so what we do is that as the corruption probability is changing, we feed into our model this corruption probability, and we see if it's able to use this information to overcome the difficulty of noisy labels. What's interesting is that you see that when the corruption probability is of 0.2, our model and the original M1 plus M2 model, they perform in the same way, which makes sense because we said that we could interpret the M1 plus M2 model as modeling uncertainty being the same for all classes. So when the probability corruption is 0.2, as we chose the other probability corruption as 0.2, it's a sanity check to see that the models perform in the same way. But as soon as the corruption probabilities are different between the classes, we see that the performances in terms of accuracy on the true labels of our model is better than the one of the M1 plus M2 model. And we can interpret that as saying that the information that we gave with the Bayesian specification of a model on the error process in the labeling process is actually used to get more robust to labeling errors. All right. So do you have any questions?
So I'm curious, in general, it seems a problem to design the confusion matrix itself. Have you thought about that? In general, how would you address that?
Oh, yeah, so there is, it's actually a very interesting question. You can do it in various ways. You can do it like manually with, for instance, an expert knowledge of the field. You can say, okay, I know that there is often confusion between those 2 classes, so I'm going to put a high weight in the confusion matrix at this entry. But you can also choose to put it as a free parameter of the model and to let, throughout the optimization, to optimize on the confusion matrix.
Okay, thanks. So let's thank the speaker again.